Good morning, everyone. My name is Cass, and I'm an intern at the CFP. So, for the last 6 months, I've been working at Kew. And what I've been doing is some computational work on a family of elm trees. And I've been looking for genes that are involved in resistance to Dutch elm disease. So, I'll start by introducing you to the experimental setup, and I'll give you some context about where the data comes from and the techniques that we use to look at the data. So around 10 years ago, David Hurling crossbred an elm cultivar called FL493 or Almus Wingham, which you can see on the right here, with a field elm. And David was really passionate about elm trees and he really wanted to try and restore them to the landscape here in Britain. And although Wingham is considered very resistant to Dutch elm disease, it doesn't really have the classic appearance of field elms that so many people love about elms. So he brought up this large collection of saplings with the hopes of creating a tree that combines the resistance of Wingham with the appearance of field elm. And if you look at this family tree here, we have Wingham at the bottom, and Wingham is a really interesting cultivar because it's the result of a four-way admixture between four species of elms. So it contains genetic material from two European species, which you can see at the top on the right here, Ulmus glabra and Ulmus minor. And it also contains genetic material from two Asiatic species, Ulmus pumila and Ulmus willichiana. And so David tested these saplings for resistance to Dutch elm disease. So what he did was he inoculated them with a very aggressive strain of the fungus that causes Dutch elm disease. And after a period of about 12 weeks, he measured defoliation. And we considered about 25% defoliation to be the threshold at which an elm was considered susceptible to the disease. And the results were actually a very mixed bag. I think about 26 elms were considered resistant by that threshold, and about 34 were showing quite considerable signs of having succumbed to the disease. So the technique that we use to analyze the genetic data from these trees is called chromosome painting. And in our case, what that means is we look at all four elms that come from all four of those species that contribute to the genetic admixture, and we look at their chromosomes. And then we look at the chromosomes in the offspring. And looking at the chromosomes in the offspring, we try and figure out how to trace back every part of every chromosome to one of those four species, because we know that every part of the chromosome has to be inherited from one of four species. So we can paint all of the chromosomes according to that ancestry. And this is what that looks like. So these are the painted chromosomes for Wingham. And each of these bars represents two homologous chromosomes. So if you look at the bottom here, this is chromosome 1. And we have these blue regions have been traced back to Ulmus glabra, and these regions in orange have been traced back to Ulmus minor. This region here, for example, has been traced back to pumila, and this chromosome here has all been inherited from Willichiana. So we were actually able to do this for all the offspring and both of the parents and one grandparent in the Hurling cross. So it's just a very intuitive and interesting way of looking at how genetic material is passed on between generations. So, for example, Wingham is the offspring of a clone called 1038, but since 1038 was open pollinated, we don't know who the male parent of Wingham is. But if we look at the painted chromosomes for these two elms, we can see some of the genetic material in Wingham hasn't come from 1038, so it must have come from the male parent. And if you look at chromosome 9, for example, in 1038, we can see that this whole chromosome has been inherited from minor. This blue chromosome has been inherited from glabra, but in Wingham, when we look at chromosome 9, we see this green region, which has been traced back to pumila. So if we actually compare these sets of chromosomes, we can infer that the male parent of Wingham is a complex hybrid containing genetic material from all four of these species as well. And here are the painted chromosomes for some of the offspring. Here in this top right corner, we have the chromosomes of Ulmus minor at Tongue Mill. And since we have this classified as Ulmus minor, we'd actually expect all of these to be orange, but you can see some of the blue chunk, some of these blue chunks have been traced back to glabra. So that can mean one of two things. It could mean that either this tree genuinely has some genetic material which is traced back to glabra, and this is a hybridized elm, or because glabra and minor are quite closely related, it's difficult for the program we used to tell the difference between these two species in certain regions, where it doesn't really have enough information to distinguish between them. So some of these blue regions, I know it's hard to see, but they're very small and they're very wobbly. Not all of them, but that does tell you that there's quite a bit of uncertainty in those predictions. And then also, if we look at the offspring, you can see that the Asiatic ancestry has been diluted since crossing Wingham with a European elm. We can see that there's a lot more sort of blue and orange in the offspring and a bit less pink and green, which tells us the Asiatic ancestry has been diluted in these elms. So why do this analysis? What's the practical use of doing this? If we suspect that Asiatic elms are carrying resistance genes, then we can use our chromosome paintings to try and look for those genes. And we can compare the resistant elms and the susceptible elms and look for regions where we see that the resistant elms have inherited some material from the Asiatic species that hasn't been inherited in the susceptible elms. So in this slide, we can see um, on every chromosome where material has been inherited from Ulmus pumila in the resistant trees and the susceptible trees. So if we look at chromosome 9, for example, we can see that the susceptible trees have actually been the ones to inherit more material from Ulmus pumila than the resistant trees. And if we look at this next slide, this is looking at which chromosomes have sorry, this is so sorry. If we look at chromosome 7, if we look at this region here, we can see that the resistant trees have been a lot more likely to inherit genetic material from Ulmus willichiana than the susceptible trees. And that tells us that we could potentially find a gene or a few genes in this region which are conferring resistance to Dutch elm disease. But what's important to bear in mind here is that this is a region that actually represents about 35 million base pairs. So although we've been able to narrow down our search from 1.8 billion base pairs to 35 million base pairs, it still leaves us a lot of work to do in terms of trying to find very specific genes. But it's a really good start for future research. And something else that we looked at as well was how do disease symptoms correlate with overall ancestry proportions for each species? And by global ancestry proportions, we just mean how much genetic material has been inherited from each species overall. So for the Hurling cross, having one parent, which is Ulmus minor, means that the global ancestry proportions are at least 50%. And in this case, they're actually closer to around 70% on average because of some of the Ulmus minor content in Wingham. But interestingly, we didn't really find a strong relationship between disease symptoms and global ancestry proportions. Actually, if you look at these plots, ancestry from pumila, for example, is slightly correlated with more defoliation, and ancestry from minor is slightly correlated with less defoliation, which is sort of the inverse of what you would expect because we know that pumila is resistant to the disease and minor is susceptible. So from this, it kind of suggests to us that disease resistance seems to be conferred by a very small number of genes rather than being something that is influenced by a really large number of genetic factors. So that's just a really quick overview of the work that I've been doing. Thank you so much for listening. And I'm very grateful to David and Fergus for providing you with a really rare and invaluable resource for elm research.